Hello everyone. In this video, we are going to talk about some basics in microbiology to introduce you to this subject. So, what is microbiology? Microbiology is a study of microscopic organisms such as bacteria, virus, fungi, and protozoa. Most of these microorganisms are beneficial to us. For example, humans have normal microbiome that helps in digestion. In plants, bacteria help in nitrogen fixation, etc. These microorganisms are also used in industries. Generally, we divide these bacteria, viruses, and fungi according to their characteristics. So, in bacteria, we use gram stain, either positive or negative, to categorize them. We will get into detail about gram stain soon. In viruses, we depend on their genomes, DNA or RNA, whether it's single stranded or double stranded. In fungi, they are divided into molds, which are multicellular, or yeast, which are unicellular. We also have hierarchy of classification to put organisms into categories. So from the highest to the lowest, it's domain, kingdom, phylum, class, order, family, genus, and species. From the top, we have three domains on this planet: bacteria, archaea, and eukarya. Both bacteria and archaea are considered as prokaryotes, and eukarya are considered as eukaryotes. The main difference between prokaryotes and eukaryotes are absence of cytoplasmic structures like the mitochondria, Golgi apparatus, endoplasmic reticulum in prokaryotes. After that, we go to the kingdom. Both bacteria and archaea have only single kingdom, which are bacteria and archaea respectively. And then we have eukarya, and it has four kingdoms: protists, fungi, plants, and animals. Now, focusing on bacteria, their name is composed of two words. First one refers to the name of genus, and second refers to the species. We will take Staphylococcus aureus as an example. So, Staphylococcus is the genus and aureus is the species. Also, the name of the microorganisms might refer to its shape. Like in our example, Staphylo means a bunch of grapes or a cluster, and cocci means round cells. Aureus means it looks like gold in color when we culture. Other than cocci, the round cells, we have other shapes too, such as bacillus, which are rod-shaped cells. Example, Bacillus anthracis, which is the causative agent of anthrax. Spirilla, which are spiral-shaped cells. Vibrio, which are comma-shaped cells. Example, Vibrio cholerae, which is the causative agent of cholera. These were some basic shapes of bacteria. Other than these, we have spirochete, which are curved cells. Diplococci, diplo means two, and cocci of course means round. So diplococci are basically two cocci cells or two round cells. Cocobacilli, mixture of both rod and round shaped cells. Streptococci, cocci in chains. Staphylococci, large cocci in irregular clusters. Tetrads, tetra means four, so it is the group of four cocci. Next, we are going to look at the structure of bacteria. As bacteria are considered cells, that means they have genetic material, a cell membrane, and cytoplasm. In addition, they have a cell wall composed of peptidoglycan, a flagella, which is used for locomotion, a pili, which is used for attachment, and a plasmid, which is an extra chromosome of genetic material, so it's not a part of DNA. It also has Glycocalyx, which is used for protection. The Glycocalyx can be in the form of capsule, which is neatly organized and protects the bacterium from phagocytes or as a biofilm, which is an unorganized loose structure yet important for attachment. There are also porins. These porins are transmembrane proteins which selectively allow some molecules to pass the cytosol of the bacteria. Porins can mediate antibiotic resistance by inhibiting some antibiotics entering the bacteria. They also have mesosomes. Mesosomes are equivalent to mitochondria in eukaryotes. Remember, bacteria doesn't have mitochondria, so it depends on these mesosomes for respiration. Mesosomes are formed by invaginations of a plasma membrane into the cytosol. Now moving on to the gram staining, we can classify bacteria as either gram positive or gram negative, and that depends on the constituents of the cell wall and the ability to react with the gram stain. To classify them, gram positive reacts with gram stain and gives blue color, while gram negative reacts with gram stain and gives red or pink color. If we look at the membranes of both gram positive and gram negative bacteria, from the inner layer we can see that both of them have plasma membrane. However, from the outside is where the difference is. In gram positive, it has thick peptidoglycan layer. It has two teichoic and lipoteichoic acids. In gram negative bacteria and outer to the plasma membrane, we have a thin layer of peptidoglycan, and then we have an outer membrane composed of phospholipids that has trans proteins and lipoproteins embedded into it. And the last layer in a gram negative bacteria is a polysaccharide. The lipopolysaccharide layer is composed of three structures: O antigen, core polysaccharide, and lipid A, which is also known as endotoxin. So we can conclude that endotoxins are present only in gram-negative bacteria. These endotoxins are antigenic, which means they can elicit immune responses causing symptoms like fever, weakness, aches, or shock to the patient. So that's it for the intro. Hope you enjoyed it. Thank you for watching.